Thank you very much, and thank you for selecting this abstract. Yeah, I'm quite new to the polyphenols world. It's kind of the work of the last 1.5 years, but today I try to guide myself and you into this world and into how omics contribution can help us understand more how polyphenols can be of great help for our health. This is actually one of the projects that we have in the Human Nutrition Unit at the University of Parma. This was financed by the European Research Council, led by Professor Pedro Mena. And actually, we are trying to understand how polyphenol metabolite can actually have an impact on health and how they can be beneficial for cardiometabolic health, starting from healthy individuals and then moving into individuals affected by non-communicable diseases. So I was quite new to the world of polyphenols, and I think that since 1 year, I'm trying to understand what polyphenols are. Basically, these are plant bioactives that are found in plant-based food. They just have one or more attached phenyl group to their molecules, and they're basically widespread in different classes in all as you can see here in the plot, in basically all the plant-based food that we normally eat. So they're thought to be actually being very different across individuals, having a high inter-individual variability, and at the same time having health-promoting effects in humans and also in plants. And when I was starting working with that, coming from human genomics, I was like, do we have a reference when I'm investigating omics analysis to understand something more on polyphenols? And actually, we didn't have. We just have this kind of list of genes that were related to polyphenols, some coming from in vitro evidences, some other coming from inferred functionalities, but there were no pool data that we can use to actually understand more on the genomics part. So we started doing some network analysis. This is an analysis made by string in which we can find actually that among the 121 nodes that we identified, meaning genes, so proteins, there were a high interconnectivity between them. And we can show actually how the biological complexity in human genomics related to polyphenolic metabolisms and bioavailability was actually available. So as you can see in the plot, there was a strong interconnectivity, and not everything was connected, but we can find some hotspot in which these genes were actually related. Going over that, we were actually asking, okay, we can see connections, but at the same time, we want to understand the function of these genes. And doing some clustering, for example, so K-means clustering, we identified that these genes were actually related to metabolisms and related to how the metabolic bioavailability and the metabolic pathway can actually help us understand more. As you can see, we find something related to cellular glucuronidation, key process for the polyphenols, metabolisms. Some were related more to families of genes, such as the mucins, xenobiotic transport, bile acids, recycling. So all these some sort of processes that were actually related to polyphenols, metabolisms, and bioavailability. We were actually pretty happy. Can be actually something that was straightforward to what we were investigating on, but we actually found some sort of a reference that we can use for further analysis. So starting with this very preliminary, we put hands-on to this project, the Predict Care project, that was actually aiming at investigating the health effect in cardiometabolic health and cardiometabolic status in healthy individuals and in diseased individuals. We actually have this population coming from the northern part of Italy, mostly from the Parma region, the Parma area and the Emilia-Romagna region, in which we used and we collected several biological samples to identify metabolites, quantify metabolites, but also to investigate different omics layer and different data coming from surveys and other informations. This is our core. Basically, we assessed 400 individuals, and then at the end of the day, we were able to actually have the information and all the data collected for 297 individuals. These are some stats. I think I'm not going into that, but I mean, we hope that the paper will be out soon, so hope you can read them. They were basically having 40 years old, and they equally distributed among sexes, and they were actually all pretty healthy in terms of cardiometabolic health. We collected some sort of kind of big information regarding anthropometric measurements, macro and micronutrients, food groups, polyphenols intake, and dietary pattern. Then we also collected information regarding cardiometabolic health status. Through blood samples, we collected cardiometabolic markers and also blood to perform genotyping. Through the stool, we collected information, basically biological data, to perform metagenomics investigation. And through the urine, after a washout of polyphenols, we actually supplemented with tablets made of a strong presence, a strong abundance of polyphenols, actually to understand the urine metabolome and actually the production and the variability of phenolics metabolites into our cohort. Since the nutritional investigation mostly are around metabolomics and metagenomic study, I'll start presenting today metabolites and metabotypes, basically our metabolomics investigation. This was performed by all these beautiful people in the lab. Basically, we take the urine samples and we try to understand which and how much the metabolites were present coming from phenolics compounds in our samples. We monitor 243 metabolites, identified 213, and at the end of the day, we were able to retain 208 metabolites. We had all the levels for all our 297 samples in our collection. However, we started pulling together this data in terms of metabotypes, meaning that we try to pull together this information, creating binary or clusters, binary phenotypes or clusters. For these metabotypes, we actually investigated some sort of 1,500 different models, combining different data pretreatment approaches, different dimensionality reduction techniques, as well as clustering algorithms. And at the end of the day, we found out that the long-word P transformation, no centering and no scaling, combined with the UMAP dimensionality reduction, and ending with the k-means clustering was actually the most performant for identifying these metabotypes. We were able actually to identify two different metabotypes. They are not very separate clusters. However, we can see that there's some sort of a trend between these two metabotypes. We call them at the moment metabotype 1 and metabotype 2, and these were actually characterized by different phenolic abundance in the urine samples, and we inferred that the one having low phenolic production were actually more related to high cardiometabolic risk, and also the one being from the metabotype 2 in green, having a high phenolic production and high phenolic abundance were actually more related to a low cardiometabolic risk, as actually the literature was kind of suggesting. Moving after that, we looked into metagenomics, so to our fecal samples. We perform some sort of standard metagenomics workflow, basically investigating the ITS, internal transcribed spacer sequencing, to identify kind of 100 families, almost 250 genera, and 483 species that we try to understand whether these species can actually be specific to one of the two metabotypes. However, when investigating the diversity inside our metabolites, we weren't able to identify significant results between alpha and beta diversity. So we tried to understand something a little more on the metagenomic side, and we performed some sort of differential analysis using log-fold changes, finding out that there were some sort of trends for metabolite-specific species related to polyphenols, bioavailability, metabolisms, and biotransformation, being here in red, such as the flavonifactor plausi and the pseudoflavonifactor species. However, these were actually some sort of a trend, so we are still investigating more and digging in to understand something more about the microbial species living in our organisms and in our samples. After that, we start looking into genomics of our individuals. We perform a genomics workflow, basically based on the Illumina global screening array genotyping, moving then to expand our understanding on imputed data, and then performing genome-wide association studies, a computational functional analysis. And we identified from this cluster 297 individuals divided almost equally in 128 in metabotype 1 and 167 metabotype 2 that we addressed genetically, performing a genome-wide association studies. Since being one of the very first genome-wide association studies, we actually decided to lower our threshold, being some sort of exploratory GWAS, and using the common suggestive threshold to 10 to the power of -5 as our genome-wide threshold and a -3 as a suggestive threshold. We find that some of the peaks were actually over our new genome-wide threshold, and some others were actually between these candidate regions. To understand more about these genome-wide association studies, we performed with Fumejiwas some functional mapping and annotation analysis, identifying 3 risk loci that were over the 10 to the minus 5, and also identifying that the variants related to these genes, as we can normally see in complex diseases and complex traits, were mostly intronic and intergenic. To get something more, we try to understand basically on the only loci having one single gene inside it, something more about its genetics, and performing some functional analysis and using public available data on chromatin accessibility, as well as QTLs. We found out that there was some association related to the TSHR gene, the T-rate stimulating hormone receptors. And since there was something pointed at that, and this was an unprecedented association reported for polyphenols, we are actually trying to understand something more about that. And basically, we found out that these genes and the receptor coded by these genes is actually a structural affinity with basically phenolic structure. So we are now trying to understand something more about that in order to see whether these genes plays a role, or rather we are just seeing some associations related to the genes but being actually a proxy to other signals. So we then look also for the genes that we put into the network to understand whether some genes of interest were actually significant. However, we did not identify very strong associations. They were in between the 2 zones, so basically in the suggested zones. And they were mostly related to the UGT1A complex, so related to glucuronosyl transferases, sulfotransferases, sulfatases, and ABC transports. These were just suggestive signals, so we were trying to understand whether we are having a small sample sizes rather than we have to dig in more with other approaches to understand more about these genetic variants into the genes of interest. However, we find out that there might be some possible regional effects when investigating public available gene sets related to the association of these genes. When it comes, however, to understand something more about the association to the cardiometabolic health, we didn't find any genetic associations. So we start collecting and analyzing the information regarding cardiometabolic biomarkers as well as cardiometabolic information. Our hypothesis was that the metabolite 1 was associated with low cardiometabolic health and the metabolite 2 with high cardiometabolic health. However, when trying to correlate this classification with either cardiometabolic biomarkers or cardiometabolic risk scores, we didn't find any significant correlations. So to try to understand more and try to validate our hypothesis, as was observed by other research, we actually try to use the polygenic score catalog in order to extract all the polygenic scores, apply them to our population, and made some statistical analysis to understand something more about any possible genetic liability that was unseen in our genomic associations. We extracted 3,295 scores. We computed some pairwise t-tests and some pairwise statistical analysis to compare the 2 metabotypes. And then at the end of the day, we find out 180 significant polygenic risk scores that were actually groupable in 20 groups, as you can see some example in the table. When just to present a couple of actually examples, we found some associations significant, for example, for coronary artery diseases. You can see in blue the metabotype 1, and you can see in red the metabotype, you can see in blue the metabotype 2, and in red the metabotype 1, and of course also for the LDL cholesterols. And we can see that genetically there might be some differences between some sort of undetected genetic liability that might differ these kind of metabotypes. So we are now actually wondering of a possible influence of the genetic background that is actually is falling under the genetic complexity and the unseenable data that we have so far. After all of that, we were trying to understand something more about multi-omics. I know I speak fast, so I didn't went in deep into that, but actually we tried to perform using the most common Mixomics Diablo tool about some B correlation or correlation of the whole omics that we had. Basically, we tried to find out whether multiple signals coming from different omics can actually explain the phenomenon that we are observing. But yeah, not going into that, but if you have time during the next poster session, my PhD student, Lucia, is here in the room, is presenting a poster, and I think is more than happy to explain you what are the last findings about that. So in conclusion, basically using metabolomics, we identified these two distinct metabolites. However, we are now trying to investigate the different level of these 208 identified metabolites to understand whether some specific metabolites can actually be of interest when investigating cardiometabolic impact of polyphenols. Moreover, we didn't find some strong significant association in the microbiome. We are actually trying to understand whether this was related to the techniques, so the ITS sequencing, and we are now moving into whole genome shotgun metagenomics to get something more and also to understand more about the functionality of possible bacterial genes related to polyphenols. And when it comes to genetic association, basically we just see suggested association involved basically in unprecedented markers and genomic risk loci, but at the same time we just wonder what about the genes of interest that are related to polyphenols, bioavailability, and metabolisms. So we are now trying to put together the data and basically to understand something more about that. And basically we are just trying to look into the nose, since in the human nutrition and in nutritional investigations, this study are actually quite underperformed. We are trying to gain in to make collaboration network and to try to understand something more in other cohorts. Basically, this is where we are trying to go, so applying advanced model coming from different omics layers to produce data guidelines as well as personalized treatments. And now we are trying to applying everything, not just to a healthy population, but also to disease populations, such as the one in the precare diet, having cardiometabolic, being a high cardiometabolic risk, but also in the mentoring for health, investigating non-communicable diseases, as well as population that are actually affected by cancer, such as lung cancer. I want to thank you all for your attention. I think in this presentation, we spare some time for some question, or we resume time with the schedule. And of course, I want to thank all the groups. I mean, it's not just my work, it's the group of a big team and big partners, as you can see here. And yeah, I'm more than happy to welcome any of your questions.
Thank you for the wonderful presentation. We have time for a few questions.
Hi, thank you for a very interesting talk. I was wondering if some of the genetic variants that you identified have also been found in long-lived populations, and if that maybe explains longevity. And I have a second question also, if I may. Why do you think there's genetic variation for this trait in a human population?
Well, regarding, I think you mentioned, thank you for the question, though. I think you mentioned for the older population, right? Because I can't hear properly the adjective on the first question.
The first question was relating to whether some of the variants that you identified explain longevity differences, like differences in lifespan and aging.
Yeah, I think that we looked into that. We look into association and we look into reported papers about the variants that we identified. However, most of the time we didn't find any function related to these variants. No, we didn't find any function related to association to aging. So basically, we didn't know why this genetics variants pop up. Then may be false positive. Then may be something that we don't know, but we are still trying to dig into that. On the second, on why we are doing genomics, basically.
It was, why is there genetic variation for this trait?
Yeah, why we think there's genetic variation? I think that there's not something related to exonic variants, if I may, something more related to probably regulatory variants. And this is the first layer that we're trying to investigate. So basically variants in the DNA sequence. We're now moving into like epigenetics and transcriptomics to see whether there's something related to some more effective biological actors for that. So this was just the first layer to understand whether there would be some genetic associations. Actually, it seems not to, or seems that it might be empowered. So yeah, we're still figuring out whether there might be. Thank you.
I just hope it's not lost on everyone how amazing it is that you were able to pull off that cohort and collecting all those samples. That's remarkable. But quick
Thanks.
question: to what extent can we take the phenol metabolites
Yep.
to actually be, either consumed, produced by human versus interaction with microbe, etc. So we know where the polyphenol is coming from, or the metabolite of such is.
Like we assessed that using multiple surveys from food habits at the first time. So we try to understand whether there were some food consumption related to having phenolic metabolites in that. Before doing the analysis, we perform a polyphenol washout. So basically we try to set that to 0. We know that that's impossible. After that, we created specific tablets rich in polyphenol that we can monitor. And then using metabolomics, we try to understand whether the polyphenols that we gave to the volunteers were actually present in different amount. Then we perform metabolomics investigation by different techniques. We have a huge lab for that. I think, basically by UPLC, IMS, HRMS, and UPLC, QQQ, MSMS, using a lot of standards to understand the presence in the urine. And that at the moment, we're trying to see whether all the metabolites that we can detect but are not produced biologically by humans are actually accessible and addressable to the metagenomics, so to the microbes, rather than are coming from other stuff. For that part, we are now producing metagenomics data in order to address that more related to function. So at the moment, we do not have the answer because the ITS just show us basically the taxonomic assignation. But yeah, I think that's our next step, and I think we can let you know shortly about that.
Okay, thank you.
Thank you.